In this simulation, a colony of E. coli bacteria is grown in a chemostat with a food source of gradually decreasing concentration. Each bacterium has two genes, a metabolic rate and a mutation rate. Each time a cell divides, the daughter cell's genes are mutated according to the mutation rate. The metabolic rate and mutation rate of all cells in the population are shown in the graph to the right. The red line indicates the maximum metabolic rate the current food supply will support. As the amount of food declines, cells with a high metabolic rate starve and die, while cells with a low metabolism survive and reproduce. While it is intuitively obvious that natural selection favors cells with a lower metabolism, the graph shows that the population also evolves toward a higher mutation rate. When the amount of food is dramatically increased, this highly variable, highly evolvable population evolves to take advantage of the situation. This demonstrates that natural selection can favor a decrease in the fidelity with which an organism copies its genetic material. Imagine the opposite, an organism that had the ability to copy its genes without error. The evolution of such an organism would be frozen, and it would be unable to change in response to a changing environment. Such an organism would likely be outcompeted and go extinct. In this simulation, the same initial E. coli cell is placed in a constant, abundant food source. The cells quickly evolve a higher metabolism to take full advantage of the available food. On a much longer time scale, they also evolve a better genetic copy machine, and there is much less variation in the population. Such a population is well suited to the conditions it evolved in, but when subjected to the same declining food source as the first colony, these bacteria cannot evolve quickly enough to avoid extinction through starvation. This GROW simulation was written in GROW. GROW! What can you grow?